familiar with face recognition, so AI must be, of course, must be able to do image recognition. And often we related AI to some robots, so AI must be able to actually drive a robotic machine to do whatever magical act. And also, we humans sometimes do planning, plan things ahead. We also want AI to do these kind of things. So we actually has achieved a lot of things. So these are some things that I think all of you should already have already know. So I don't have to mention in more details. But rather we still need to answer this question, right? So what is an AI? Talking about artificial intelligence, we have to look at human intelligence, and then so that we can understand the AI. Okay, so we take this example, the ECG. So the when a doctor see this ECG, the doctor may react with some diagnostic decision about whether this is abnormal or it is okay, right? But for our commoners, normally, because we are not trained for recognition of these kind of things, we may just be wondering, no, what that means, right? So what that actually tells us. So we want an AI to act as a doctor. So the question is, how can AI act as a doctor to read this kind of ECG data, right? So in order to be able to understand AI, we need to understand the difference between the doctor and a commoner. After all, the doctor has the knowledge to make a right diagnosis, right? So fundamentally, we need to look at the what's inside a doctor's brain and what's inside a commoner's brain. After all, it is the brain of a human that actually make that kind of intelligent decision, right? So fundamentally, our brains are composed of many neurons interconnected with each other, right? So in a doctor's brain, when the doctor see a ECG image and her brain actually reacts with the firing up of some neurons. So there's this kind of neuron activation pattern. So it is this kind of pattern actually make a doctor to actually decide whether or not it is normal or abnormal, right? And in contrast, our commoner's brain also has some kind of activation pattern in this kind of network of neurons. The patterns obviously are different from a doctor's pattern because uh, if a doctor notices something serious, her brain must be very alert for, right? But if we have no idea about that, we just wondering without a really serious alertment. So that's the difference that can be reflected in a pattern of neuron activation, the activation patterns. So a AI, so now we can build an AI to try to imitate the brain, the network of neurons of a doctor. And we wanted our network of neurons, artificial neurons, or network of nodes to be in such a way that it has also some kind of activation pattern. One pattern corresponds to abnormal situation, right? The other corresponds to healthy, normal situation. If we are able to do that, we can build a artificial intelligence that really imitate a human intelligence. That's how a artificial intelligence machine was perceived. So it is essentially some kind of network of nodes, and each node has a certain activation condition, activation state, and it is the collective pattern of all these nodes, activation uh, state of the nodes that are really uh, make the network to be able to make some kind of judgment. So this is how AI actually uh, is built, was built. But we have not solved these fundamental questions, how we can develop such a artificial network, or we call it artificial neural network. So how can we make this kind of connections of nodes to be of different activation states? So essentially, we have to rely on mathematics. So it is the mathematical relationship that interconnects one node with another that makes this pattern possible. So in actually in today's lecture and also in the follow-up lectures, we will repeatedly look at how we can build a mathematical relationship. How can we develop this kind of complicated networks to actually make this pattern possible. Okay. Essentially, the mathematics works in such a way that on the left-hand side, we have the input of any kind of data, be it be a ECG, a image, or some sort of whatever kind of data. So essentially, all these kind of data is eventually converted into some vectors, some matrices, and then the vector and the matrix is going to be input into this network of nodes. And then we use the mathematical relationships to actually make this network to work in such a way that uh, when we input any data, uh, some may be normal, some may be abnormal, and then the data, when the data goes through this network, eventually we arrive at a new bright space. It's called a deep lightened space in such a way that the normal data and the abnormal data are nicely separated into two parts with a very simple borderline to separate them. In this case, it is a straight line. Of course, in reality, it is of a high dimensional space. So essentially, it is a hyperplane that separates the abnormal data from the normal data. If we can do that, if an AI can do that, of course, uh, diagnosis of a disease becomes simpler. You can simply look at whether or not a particular patient is on the abnormal side or the normal side. That's how AI works. Okay. So now, from what we can understand, AI must be some kind of network. So this network can be very simple, could also be very complicated. So this is analogous to a some kind of a circuit. We can have a very simple circuit, just a battery connected, of course, with a switch connected to a bulb, light bulb, right? And of course, if we want to the circuit to do more magical things, and then we have to have a more complex network of circuits, it becomes a integrated circuit, right? And if we wanted it to be more complex, essentially we have a specialized chip, many different chips. For instance, in a computer, in the GPU machine, server, and so on and so forth. So our artificial neural network actually goes through the same kind of development stage. Initially, we have a very simple network, and then we have a more complex network. It is a lot more look like a integrated circuit. But nowadays, what we actually see is not this kind of nodes connected by some lines. Rather, we see the kind of network is more like a composition of many blocks. Each block you can imagine, each block is simply some kind of a chip that contains inside it actually contains many different nodes connected by the lines or something like that. Okay, so now we can look at the image recognition AI. Okay, so an image recognition AI, so is normally is the one on the top. You can see essentially is some kind of composition of many different chips. Okay, so this kind of chip architecture is very good at something to actually capture the features of an image. Okay, so an image, you can imagine an image is something that has a very unique uh, characteristics. For instance, if we look at the image on the left-hand side, we see the lower part of the image is actually grassland, right? And then on the top, you see the light blue sky with some clouds, and in the middle you see the water with the boats, right? So when we look at the image, we do notice that on the lower part, it's dominated by the green, maybe with some soil, right? On the top is the sky with some clouds. In the middle, you have a boat and the water. So you see that an image has this kind of characteristics. It has kind of these so-called local features, like the grass on the lower part, sky, light blue sky on the top part, right? And it also has a combination of local features like the boat inside the water, like the soil surrounding soil in between the among the grass, right? These kind of things. Okay, so our this kind of image recognition AI is designed in such a way that it is very good at this kind of focused scanning of local features and local feature combination. So it is scanning for local features and their combinations. So this kind of architecture is called CNN. Okay, we will come back to CNN in the later lectures, but right now you just need to know we have this kind of specialized chip that are very good at capturing or scanning the local features of the data or the combination of the local features. This is called SIN, very good at image recognition. Okay. Now, what about language learning? Okay, it's a kind of more complicated. So we already mentioned that AI must be able to do whatever our human when we read something, right? If we read a question, we can write down an answer. If we read the information, we can have this kind of judgment whether it's good or not. Or if we read one language, we may be able to translate it into another language, right? So this kind of capability must be handled or must be processed by this kind of language learning AI. So how can a language learning AI to do these kinds of tasks, right? Obviously, the CNN architecture no longer works because language is very different. These texts, they are very different from images. Images, you have so-called local features. What about the language? This example shows, right? We have this kind of sentence that many kids know, right? Old MacDonald had a farm, right? So when we read these kind of sentences, we notice that the words behind the words, some words they somehow have some kind of relationship, either logical relationship or the relationship that emphasize some words or make some words more meaningful, more standing out, and so on and so forth, right? And so this kind of before, after uh, relationships are the ones a language learning AI must be able to capture. Okay, so one this kind of this kind of AI is called nowadays people use transformer. Transformer is the this kind of new new type of chips that are actually actually specially designed for this kind of language learning. But a transformer uh, is not the only type of architecture or only type of chips that can do this. Actually, before transformer, there are some other AIs like RNN. It can also do this kind of learning, capturing the relationship of before, after, these kind of relationships. Uh, but why I mentioned transformer? Because transformer can read a very long sentences without being lost with some information. What about RN? RN can only read short sentence. If a sentence gets longer, an RNA architecture can easily forget the earlier words, only be able to remember the later words. But for transformer, it's no longer a problem. You can have a very long sentences. Now actually a very long text. It's no longer sentence. It's a very long text. Actually, it can be as long as several hundred K words. So it's very long text that transformer can really handle. So here is the actual architecture of transformer. We will come back to the transformer architecture later on in a specific lecture, but today we just go over it to have for you to have understanding what an AI is. Now we come to the second question. How does an AI work? So we take this example. So this is an example particularly for the earlier AIs, okay? So in this example, we have 6 objects. Some objects are cups, some objects are opposite, non-cups, okay? So we could have some kind of text to describe each object. Now we can actually extract some specific information from the text about each object, like the color, the material type, whether or not it has a handle, whether or not it is so heavy it cannot be liftable, so whether or not it's liftable, meaning that the liftability of the object, and also the last thing that we extract is about the shape of the object, particularly whether it has a concave shape or not. So essentially, we extract these 5 features from some kind of text, of course, and we could simply give the text rather than the extracted information. But as I mentioned that this is a example for the earlier AI. That means the AI before deep learning. So regarding what kind of AI, we will talk about it slightly later on. Okay, so now we look back this example, and we can see that these objects can have different colors, different material types, and so on and so forth. But we do notice that if this object is liftable, and if this object has a concave shape, and based on this table, we know that it is a cup. Okay, This is what our human brain can actually draw some kind of pattern, right? We notice this pattern through our eyes, processed by our brains. Now the question is, how can we develop AI to do the same thing? Uh, I mentioned earlier that we must have some kind of networks, but networks, as I mentioned, that it has to be realized through some kind of mathematical relationship. Otherwise, a network is still some kind of concept, right? Some kind of imaginary type of network without really something that you can actually make it work, uh, either using computer or using any other form. For instance, sometimes you may be able to really write down the equation, simply solve the equation rather than using some numerical machine, right? But here in our example, we really want to develop a mathematical model simply from this table. How can we do that? Okay, so that's the question. Okay, when we look at this table, of course, we notice that these are all descriptions. Essentially, they are composed of some words. But these kind of words may be they may be meaningful to our brain, but how can it be meaningful to a computer? So that's the question. So now we still have to go to the fundamental, just like when we introduce artificial intelligence, we have to look at the fundamentals of human brain, those network of neurons, right? So the fundamental thing about a computer is that a computer is a digital machine, right? Essentially 0101, those digital machine. Okay, so now what we need to do is that we need to digitize this table before we can actually send the information to the computer. So the first step is to digitize it. Okay. Okay. So we digitize it by converting the previous table into this one. For simplicity, I only focused on three features, whether or not it has a handle, whether or not it is liftable, whether or not it has a concave shape. With the handle, you can assign a value 1. Without, it is value 0. Liftable 1, non-liftable 0. Concave 1, non-concave 0. Right? So we have this table. Okay. Uh, if you are good at mathematics, and when you look at each object, A, B, C, D, E, F, G, right? Each object, you notice each object is composed by three values. It is a three-dimensional space vector, right? So if you are good at mathematics, so now we convert the objects. We first digitize the object. Second, we convert the object into vectors called the feature vectors because we really first draw these features before we can actually digitize it, before we can actually generate the vectors. Okay, so if we have these vectors, we immediately know it must correspond, any vector must correspond to a particular position in the high dimensional space. In this case, it is the three-dimensional space because we can easily draw a three-dimensional space distribution, right? We have a three-dimensional XYZ coordinates, and we can simply draw the each vector point on this three-dimensional space. Okay, remember that some points are positive cups, some points are negative non-cups, right? So now we have some kind of distribution about certain points are the positive ones, certain points are the negative ones. So the question is, how can we draw a border to actually separate the positive from the negative? So this is the question. Okay, so without because remember that I mentioned that we must have a mathematical relationship. We can't simply have a vector and then display the vector in a three-dimensional space or high dimensional space. What we need is a mathematical relationship. Now we try to come up with that kind of mathematical relationship. Otherwise, we cannot build a AI model. Okay. So we now convert into a more relevant example. So it is about the medical example, right? So we have on the one side the cancer patients, on the other side the healthy person. Okay. So of course, we make it simpler. We assume that cancer patients are easily separable from healthy ones. Of course, if it is not the case, and we can do that later. We first look at this simple, easier example, and also we make it easier by only look at a two-dimensional space. X coordinates correspond to one gene, and then the Y coordinates correspond to another gene. We have that means we use two genes of each person to actually draw this kind of distribution. Okay, the location of the patient. And again, using our brain, we can tell that we can design a coordinate system like this one. We make the origin of the XY coordinates to be in the middle between the cancer and the healthy. And this way, we can develop some kind of simple rules to separate the cancer from the healthy. So we can design this kind of rules. The rules is such that if gene one corresponds to the gene on the x-axis, gene two corresponds to the gene on the y-axis. Okay, so our simple rule is like this: gene one, if gene one less than 0, gene two and gene two less than 0, it must be healthy. If both are greater than 0, it must be cancer, right? So we seem to have some kind of a mathematical model. Of course, this is a mathematical relationship. Of course, this is a rule-based relationship. But now we have a problem. What if we don't simply have a few genes? What if we have 5,000 genes? We have a rule with 5,000 lines. Obviously, it is not a simplest mathematical model. In mathematics, what we want is a relationship that can be expressed in a very simple way, in a simplest way. Okay, so that's what we look for. That's what mathematics is really about. So the question is, what is the simplest way to separate the patient from the healthy? So that's the question. We look back at the distribution. We know that actually the most simple way must be like just a simple, you draw a straight line to separate the cancer from the healthy. So now the question becomes, how can we develop a mathematical model to draw that line? So actually all the AIs, uh, in the last step of all the AIs, like a transformer, like a CNN, like the machine learning, the earlier AIs, they always come to this last step to draw a line to separate the positive from the negative. Okay, so the question is, how can we draw that line? Okay. First, we need to have this mathematical equation for a line. So that's easier. X1 W1 plus X2 W2 equals to T. So that's a linear equation in 2D for a straight line. And then we can extend it to 3D to n-dimensional space. To save the time, I just go quickly through it. Okay. With this kind of linear mathematical equations, we could build a network uh, in such a way that for instance, if we input the data x1, x2, x3 from the left-hand side, and then we use a weight to actually adjust the input data, uh, just like this linear mathematical equation, x1 w1 plus x2 w2, right? So the wx as the weightage as a weight. So we have this weightage. So it is a weighted sum of x1, x2, and x3 that actually uh, connects the node, the next node with the three previous nodes. So this kind of weighted sum is simply the mathematical relationship, the linear equation we really want to develop. Oh, okay, so now we can see that we could actually develop a network that can simply reflect this kind of mathematical equation. Of course, we have not really determined the true which particular straight line must be chosen as the border. So that means that we have not determined what kind of parameters W1, W2, and the T is. So that's the task of training of the AI to determine the weights. Of course, in this particular example, I already put in the final answer. The W1, W2, W3, the value is already fixed. So this way, okay. So we can make this network even more complex. So after you have this weighted sum, you have that linear relationship, we can make a twist to actually make a nonlinear function, just as if our brain uh, in order for a neuron to be in an excited state, there must be a threshold. So we could also introduce some threshold nonlinear function. So that we explain that in the subsequent lecture rather than here. Okay. So now to summarize, we have this simple mathematical equation to determine whether a particular input data is on the healthy side or on the abnormal side. Okay, so that's the how it works. But we come to the next question: what if a distribution is not simply linearly separable? That means the healthy and the abnormal ones, they are not easily separated by a straight line, just like the case in the lower case, right? So in this case, we simply need a more complex network. For the simple one, as I mentioned, a weighted sum is enough. The top one, weighted sum is enough. But for the bottom one, a simple weighted sum is not enough. What we need to do is that we need to have a deeper processing of the information. So essentially, we start with x1, x2. We have weighted sum goes to the next the top node of the next level, and then we can have another weighted sum goes to the middle node of the next level, and the weighted sum must have a different weightage than the one on the top, and then we have a third one. So we can have three different weighted sum. We arrive at three different values. We can rename these three values as y1, y2, y3, and then we do the same trick again. We do weighted sum of y1, y2, y3, and then arrive at another level of the weighted sum, the sigma. So that kind of level actually gives us this nonlinear borderline. Okay, we can make this network even more and more complex. We can have more and more complex borderlines. So that's how it works. Okay, so so I quickly go through about machine learning and the deep learning. So what's the difference? What is a machine learning? What is a deep learning? What's the difference between them? As the example I showed you earlier, okay, we draw some features of the cups, color, material type, and so on and so forth. We use those extracted features as some kind of vector, feature vector. Okay. And then we develop a mathematical model. So that mathematical model is called a machine learning process. Okay. So a machine learning model is such a thing that we need to extract the features first, and then we utilize. So normally, this kind of feature extraction is based on human knowledge. We humans extract the knowledge for the AI, and then we supply the features as a digitized vectors to the AI, just as I illustrated. So what about the deep learning? Deep learning in the deep learning, you don't have to extract the features anymore. Essentially, you just simply supplied any kind of data, be it an image, be it a language text, be it some kind of just the voice or or some kind of spectroscopic data, any kind of data you simply supply to the AI, and then the AI has to do both feature extraction and classification on its own. Obviously, if you want AI to do that, we can understand that the network must be even more complex. You need to go deeper. Just as I mentioned, you must have a for a complicated borderline, you must have you must go to the third level, even fourth and fifth level, so it becomes so-called deep learning. So that's the difference. Okay, deep learning sounds very nice, right? We don't have to draw the feature by our own. We simply let the computer, let the AI to do all the jobs. But that's a catch. That's something we have to give up. That's the one thing is the computing power. The other thing is the overfitting problem, meaning that when we have a if when we want to use a deep learning method, we have to supply the AI with a lot more data. So in this figure, it shows the performance of machine learning, deep learning methods with respect to the size of the data. The more data, it goes to the right side. Okay, so the red line is a machine learning method. Yellow line is a smaller deep learning method, and then the blue one is the larger deep learning method model. We can see that for a low number of data, the red one actually significantly outperform the blue and the yellow ones. So that means that if you have a smaller number of data, you have to use machine learning method. So that's the reason why you can see that even just in you know in in the publications of these few years in the top journals like Nature Medicine, you still see a lot of papers actually they actually use machine learning rather than deep learning methods simply because in healthcare, in medicine, sometimes you can only have just a few dozen to a few hundred patient data, clinical data. In that case, it may not be good enough to train a deep learning model. So a machine learning model still makes sense as this figure shows. As the data goes larger and larger in size, you can see that the the smaller size the deep learning model becomes more superior, and then it is the larger deep learning model becomes more superior. So you have to mindful that based on whatever data you are available, you have to consider or even you need to try which particular method is more suitable for your particular case. Okay. So regarding to the history of AI, okay, so actually because of the time of today, I will not go through because you can simply read it yourself. There are different versions of timelines of the AI history because different people or organization they actually look at the different aspects of the history of AI. I just want to and also I have listed the historical timelines for different type of AI like image deep learning, language learning, and the generative AI. But I do want to mention the history of AI that more relevant to us. That means the biomedical AI. So the AI in biomedical application actually starts with medical, with healthcare applications. It actually can go back as early as 1972. We know that AI actually it's developed actually the models those models actually developed starting from 1950s. So essentially, it's not too late after that. The first medical application of AI actually come into existence. It is regarding to the diagnosis of bacterial infections with an accuracy of almost 70% compared to 80% of doctors are specialists. But from 1998, you can see. So that's the time when gene expression technology really starts to fly. That means people can use the like microarray technology to actually measure the expression of many genes rather than individual one by one. Those kind of slow process. Because of the technology development, the people realize that why not? Because of the complex task, complex patterns of so many genes, the expression value of so many genes, people realize why not we use AI to deal with these complex problems. You can see that so the first is the unsupervised learning, and then the supervised learning, then unsupervised again, then supervised again. But it goes from very simple tasks of simple learning of gene expression pattern to the actually the disease classification, to the predict discovery of genes and also biomarkers, and also to a more specific diagnosis tasks like disease subtypes, even the new subtypes, these kind of tasks. Okay, regarding to the early history of biology, it is the same thing, but it's kind of later than in medicine. In medicine, we can see that is so it really starts from 1998. It becomes a booming days because of the gene expression technology. But in biology, it starts slightly later, 3 years later, in terms of the AI prediction of protein interactions, and then the protein structural families, and then protein functional families. Unfortunately, I was involved in protein functional family prediction back in 2003. So I'm very happy to be part of the history of the AI application in biology. Okay, in pharmaceuticals, it starts almost in the same period of time. It first starts with the pharmacokinetic prediction, understandably, because it is more complex. It involves many complex mechanisms, right? And then it goes to the toxicity prediction, and then to the collective prediction of different pharmacokinetic and toxicological properties. So I'm also happy to be part of that period of in contributing to the AI models for toxicity predictions. And also AI in the not too late. AI also is actually it's spreading from Western medicine pharmaceuticals into traditional medicine like Chinese medicine. So it has the ability for the AI recognition, AI classification of multi-herb recipes and herb pairs and so on and so forth. This kind of development. So we talk about the history. What about the future of AI? I draw this from the internet. People already We are already in the age of generative AI, right? So AI is able to actually come up with something for us to be able to actually for instance, to draw a new new art craft and so on and so forth, right? And it has also has many applications with a new device like a wearable device and so on. So what we expect next? It is so far our AI is still in a narrow stage because it is still designed for some narrow tasks. So what we expect is that we want AI to be more powerful to be able to do multitask. So we are in the stage of multitask AI. Like you already see that the large language model, they are able to mix up with the image, with voice, those kind of things, model, multitask, this kind of AI. The next thing we want is that AI must have the more capability, particularly cognitive capability. That means it can actually not just do simple tasks, but rather to more inventive type of tasks, more generative type of tasks. Uh, uh, Maybe in the 10 or 20 years time. It's called more general AI. It's no longer some kind of narrow AI. And then people thought that eventually AI, also this is still up to a question, right? Eventually AI should be more superior than human knowledge can do, human cognition capability can do. Okay, so now this is the first lecture of today. Now I actually give you the second lecture. So that's supposed to be today's formal lecture. Okay. So when we talk about AI, as the example shows, as I have mentioned a few times, we need to train the AI by supplying AI with the data. And particularly if you want to use the power of deep learning model to be able to do this so kind of multitask, multi-mode, and this kind of generative models, more intelligent, more wider applications, more capabilities, you need to have enough data to train the AI first, and then to build on more knowledgeable AI. Okay, so nowadays we are still in the age of large data model. So essentially, big data is very important, very important. Okay, so because our lectures actually focus on biomedical AI, so we want to let you know what kind of data in biomedicine that are available. Okay, before that, we need to look at the general data available for language, image, audio, AI. Okay, so the first thing is that how big the AI actually, how big data the AI actually gets into. So this actually shows you the kind of data like GPT-4, like the Grok now, it is version 4, right? Like DeepSeek, like Kimi. These are the recent models. You can see that they are with as much data as, in terms of the parameters, it is in 1.2 trillion. parameters. Okay, so essentially, it means that it needs billions more data in order to be able to train such a bigger model. So really, the big data becomes very important for developing these models. Okay. And another reason I show you this particular slide is that I want you to know what kind of computing power normally you need to train a large model. Okay, so these are the examples. You can see that 100 billion parameters is the standard, and now they go for a trillion or even more. Like Grok 4, we don't even know. Grok 3 already is 1.2 trillion. Grok 4 should be much larger than that, right? So we don't know. But we do know another trend that sometimes we may not need uh, really so big data to train a good model if we can adopt some techniques like the reinforcement learning, those kind of the kind of things or distilled type of things. So some of these techniques will be mentioned in the follow-up lectures. Okay. Now so this page shows the image data. So if you are interested in image processing AI, so these are the database you can draw a lot of different image data. Some are video, some are just pure image. Some may be human face, some may be many different objects, and so on, so forth. For audio type of learning, these are the database for audio data. Audio data. Okay. Now we go back to biomedicine. Okay. Just like the language, just like the image, big data in biomedicine is also very important. Many people mentioned it quite a long time ago, eight or even 9 years ago. Okay, and it is because of the big data that actually makes the AlphaFold works very well for protein structure prediction. AlphaFold was trained by over 100,000 protein sequences, and then together with the corresponding 3D structures, and also for medical image, uh, they are trained by over 1 billion parameters with a very large data set of 100,000 medical images with annotations. Of course, without any annotation, there are more, even more. Okay. Uh, okay, so for AI data in medicine, these are some of the online data available if you are interested in medical data. So now we look at the genomic data. Okay. Uh, Before we mention genomic data, we need to first actually give you a little bit introduction about the genome, right? So we know that our human being or any life form is composed of cells, right? So each cell has some kind of structure inside it. A particular structure is the cell nucleus, and inside the cell nucleus, there's this kind of chromosomes, right? And then chromosomes actually are composed of DNA duplex together with some proteins, these kind of things. So this DNA actually contains the gene or the genetic code of our life. Okay, so for the genomes, a genome is a complete sequence of the DNA of a life form, particularly the genes and also uh, those parts of DNA that are actually called the introns. So if you really don't know what an intron is or whatever, I suggest you to look at some fundamental biological books and to try to get more knowledge about the biology. Because of time, we can't actually go too much details in that. So this is a particularly useful database for getting the genomic data. NCBI genome database, you can see it contains many genomes in millions, those kind of things. So there are many more. So the second useful source for biomedical data is a protein sequence database. Okay, a protein sequence. We know that we mentioned about the genetic code. A protein sequence actually is a essentially a line of amino acid code. It codes the structure and the function of the protein. Okay, so if you want to get the codes of proteins, or the sequence of protein, you can actually visit the UniProt database. UniProt database actually is composed with many sub databases like Interpro it essentially contains protein family databases. Okay, of course, the main uh, data is actually the protein sequence. On the top right side, it shows how many protein sequences are provided by this database, UniProt database. Essentially, it has 50, over 506,000 protein sequences with annotated information, manually checked annotated information. It has additionally more over essentially 200, essentially 248 million protein sequences. It really has a lot of information, but those protein sequences are unprocessed by human, but rather it is processed by bioinformatic method. But still, they are useful for training the AI. Okay, the next useful one, I mentioned that actually the AlphaFold AI actually utilized is this protein structure database. Okay, it contains many protein structures. You can see that it has over 100,000 protein, actually over 148,000 protein structures, and it has some other type of structures like nucleic acids and so on and so forth. Okay, of course, with AlphaFold, we are able to have AI generated data, meaning in this particular case is the AI generated protein structures. So they are also useful for actually as some kind of supplementary data for AI training. Okay. The next useful data is the gene expression data. Okay. A gene expression, the concept of gene expression is that as I mentioned, that we have a genome of duplex DNA, right? It is composed of the introns and exons, the genes, right? So how can uh, a gene to actually be activated or expressed to be transcribed to be translated into a protein. So this is a process called the gene expression process. Okay, so in this process, the expressed gene means that this gene is actually transcribed and it can make it available for functionality. And unexpressed gene, meaning that they are silent. Those genes are still coded by DNA, but rather
Hello,
not being activated. Okay.
Professor. Professor,
Yes.
I just want to interrupt you that we have 5 minutes and you just have to wind up the lecture within 5 minutes. After that, like the students online and both in person, they want to ask some question from you.
Okay. So, okay, I no longer mention the biological background, but rather just show you the database. So the GEO database is the major one because it contains many samples of gene expression data for humans, for animals, plants, and so on and so forth. Because there are other useful gene expression database like TCGA, like biostudies and so on forth. You can read the lecture the lecture slides on your own. Okay. So another useful database is the gene ontology database. It actually provides you with the information about the functional annotation of genes. There are also database for biological pathways, KEGG or CAG pathways and the database. You can get the information. Of course, there are chemical database like PubChem. It contains many data. You can see 24 million kind of data available. And there's also this zinc database, 200 million data, and so on and so forth. Candle bioactive molecule database, it has now over 2 million data, bioactive molecule data. Drug bank for drug information, TDD for therapeutic target, and also there are some bioactive natural product database like CMOP, like TCMID, and so on and so forth. Okay. Okay, now I conclude today's lecture. So I guess we have a few minutes time for questions. Any questions?
Thank you so much, Professor, for a very different insight, particularly the biological perspective of. And yeah, here we have a lot of students, faculty
Yes.
members, and research scholars,
Mm.
and they want to ask some question from you. We both have online questions and
Okay. Mm.
in-person questions. For the
Mm.
in-person question, uh, I would like to, like anyone who want to ask question, sir, please, you just have to come here and ask from here directly. First, you introduce yourself and then just.
Assalamualaikum,
Hi.
我叫 Chan Yu Lung
Yes. Hi.
Doima. My name is Dr. Abdul Rashid. Currently, I'm working as a consultant, health initiatives, OIC COMSTECH.
Oh,
Basically, my professional is pharmacy. I'm an expert in medicines.
okay. Good.
My question is
Mm, mm.
that how you will utilize artificial intelligence to conduct clinical trials in phase 0, phase 1, phase 2, and phase 3.
That's a very good question. I think in clinical trials, there are certain kind of problems like patient recruitment, patient compliance with the clinical trial rules and like the taking of the medicine and the exams, and also the essentially the because sometimes if even if you recruit enough patients in the beginning, sometimes some people withdraw. So you need to be able to select the right group of patients. Not only they are right for the clinical trial, but also they can actually remain for the completion of the trial. So on this part, AI can help in terms of the patient profile analysis, and also in terms of essentially the classification of patients into different groups. And the second thing is that sometimes a selection of a patient for so the success of a trial depends on whether the medicine is suitable only for a smaller group of patients. If that's the case, you want to select the right smaller groups. That means you need to have some meaningful biomarkers and so on and so forth. So on this part, AI can help. Maybe you'll notice that in some recent publications for the diagnosis of classification of patients published in Nature Medicine, they frequently develop machine learning models for helping the classification of the patients for the diagnosis, prognosis, diagnosis. So these kind of applications. And also AI can help to monitoring the patient's behavior or be act as some kind of alert whether a patient may or may not be suitable or may not follow the guideline in order for the clinical trial to be valid. So these are the things I can think of.
Professor Chan, thank you very much, because other questions should also come. Thank you very much.
Okay, thank you.
Anyone who want to ask the question, please come here. You just, yes. Please introduce yourself and then.
Hello, Professor.
Hello.
Thanks for this wonderful lecture. I'm Dr. Madiha, and I'm pharmacist, and I'm working as assistant professor. And today I have a question for you. How AI can help to design a good drug with least toxicity and more efficacy, especially towards herbal medicine?
That's a very, very good question. So actually, in order for a medicine to work, it has to go through many different hurdles. The first hurdle is of course the activity, and then the next hurdle that means the potency of the drug. The next level is about the pharmacokinetics, as well as the toxicity. That's called ADME tox, right? But beyond that, there are still other hurdles. Essentially, these are you can put it in terms of off targets, in terms of sometimes called these drug-like properties, particularly the off-target effect. Off-target effect is really dependent not only on the target, the state of target tissue, but maybe the different parts of the body as well. So essentially, a drug has the effect not only on the target the tissue, but rather may affect the whole human body. If any part has some problem, it may cause serious consequences in clinical trials because the frequent failure of phase 3 trial is not only the side effect, but rather the no efficacy, meaning that it is efficacious in animal studies, but not in humans because we humans have different genetic the state or different gene expression profiles that can actually alter some kind of off-target problems. In order for AI to be able to help to design efficiently design a drug, essentially AI must be able to not only predict the effect against the tissue, not only the effect in terms of pharmacokinetics and toxicology, but also in terms of the effect on the whole body, the whole system. Essentially, it is the drug actually goes through the system rather just a particular tissue. So at the moment, we do not have an AI that covers the whole system, but eventually we can come with that. if you notice that in TCGA database, they now beginning to have the gene impression data for many, many tissues from the same person. So that's the start of the kind of effort we really we can foresee that can help us to design a better predictive power in terms of new drug discovery. Okay.
Okay, thank you very much. Thank you.
You're welcome.
Thank you. Any question from the student? I think you just have to come up. They have to come here and. So please introduce yourself and then ask.
So, Assalamu alaikum, Dr. Sahab. It's me, Ahmed Rita Bassam. I am a lecturer here in NUST University. I want to ask, like we use these large language models for text generation and other stuff for sentimental analysis. But is there something we can validate this text generated by AI?
Can you repeat again? Can you repeat?
So a lot of students and a lot of researchers are getting a lot of text generated by large language models.
Yes.
Can we have some mechanism for validations? This text is valid or generated from AI?
You mean genetics or you mean genetics, right? Because
No, I mean like the text that generated by large language models.
Generated by large language model. Text,
Yes.
you mean?
Yes.
So essentially, so the large model, they are called a pre-trained model. Essentially, it is a model that is supposed to cover everything. So if you cover everything, that means that you may not be a specialist, the top specialist for some specific thing. So that's why people have to do follow-up training for specific task, like therapeutic task, like some kind of clinical diagnosis task, like the sometimes you want to have the psychological this kind of communication or diagnosis, this kind of some people actually do this. This is the way that just like our humans, right? If you learn too many things, you are not a top specialist of one thing. But sometimes we human also have this kind of tendency. If you become the too specialist, you actually lose some general capability, and you may be viewed by the general public as just you know what I mean, right? So so what I think is that when we do the further training of the AI, we have to do both training. So we have to do double training. That means we have to on the one side, we train a specialist. On the other side, we still need to train, not in order to improve the general capability of AI, but rather to maintain the general capability. That may be possible, but we don't know. So I think it may be possible, but I really don't know yet. So that's one thing. The other thing is that when you train a specialist AI, that's something you need to do. We need to learn from our humans. When we humans train, when an expert trains somebody, it actually has some knowledge, use the knowledge to do the training rather than just this brute force training. I think for medical AI, like for instance, the protein sequence based, protein structure based, gene impression based, those kind of AI, I don't think they really have this knowledge built into the training process, but rather it's simply a brute force learning. Brute force learning may not be the efficient want to learn things. So that's something that also pharmaceutical AI is the same thing. So these are the things that we need to improve. So that's my answer.
Yes. Thank you very much. Very wonderful
welcome.
answer. I have another request. As participants, I think it will be more effective for us when you are using the slides, and maybe you can use your mouse as well. So we will know where you are focusing here. Did you get my point?
Unfortunately, so because
That's my request.
this Mac computer is very strange. There's no, there's no this jacket to actually put in the mouse. Just that's the problem. So I can't.
Thank you. Probably we have a last question from you.
Okay.
And the lady in the last row, she want to ask a question. Please introduce yourself.
Thank you for the opportunity to ask the question. And thank you, Professor, for the wonderful talk. I am Taiba Hussain from NUST University and a PhD student. My question is that can gene expression varies from population to population? I know that there's a different gene pool in different populations, but are these expressions differs in different databases? For example, I have used Alcan database, but the results are different from the wet lab. So I want to ask the question, kindly address this.
Yes, very good question. So gene expression can be measured by different techniques, like the older microarray technology and the newer ones, RNA-seq the technology. That's one thing. The other thing is that it may be measured by instrument developed by different manufacturers, and they have different kind of parameter setting and so on and so forth. So that causes some difference in the measurement, and also there are some intrinsic error in some technology like microarray. Or even in RNA-seq, that means that the impression, for instance, if you measure the same biological sample a few times, you don't even get the repeat on the same sample, and so it's caused by some was because essentially you use some kind of fluorescence type of things, there is up to some kind of error. And also there's this kind of biological variation, as you just mentioned. Biological variation is not only in terms of difference between samples, different interpersonal difference, but also in terms of dynamic difference. Even the same sample, even today and the next day, when you measure the same sample, it may be different because after all, it is a living object. It can change over time. So I think all these factors cause the difference. And then for these differences, I think the only way is that you just, so this is the reality. You need to, so that's why I mentioned that we need the knowledge, the biological knowledge to build into AI. If we know an object is dynamic, we know we should supply AI not only with the measurement of one particular time, but rather you supply the AI with the dynamic data. That can solve part of the dynamic problem. For instrument error, you need to measure the same sample using different instruments. And then the AI knows, because you can assume a sample doesn't change, it is the measurement method changes, or it is the environment or whatever adjustment of the instrument that changed. So that's called the knowledge. But unfortunately, no one do that. I think that's the problem. And also it is very costly if you do that. So now we can only rely on whatever data we have. We just hope that as we get more and more data, all these factors actually are built in, because some people use this instrument, some people use that instrument. So instrument factor maybe because these are the bias that should follow certain kind of a pattern. These patterns may be able to be captured by AI. Could be. That's
Yes,
my
I
answer.
think they should include the data from Asian labs also,
Yes,
the
yes,
databases.
yes.
Thank you, sir.
You're welcome.
Thank you so much,
You're
Professor, for
welcome.
time and energy. It's we have a short time. We have a short time. Thank you so much, Professor, for your time and energy.
Thank
We
you.
want to end this session today, and we apologize from the online who raised their hands for the question. And inshallah, we will take this question on the next lectures.
Okay. Okay.
So thank you so much. Thank you so
Thank
much
you.
for your
Thank
time.
you. Okay.
Thank you.
Okay. Okay. Bye-bye.
Share. One, Professor Chan.